My research is mostly focused on molecular imaging and the non-viral gene therapy. So as you know, DNA, RNA, and the proteins are the three major blocks of our life. DNA encoded genes, and the RNA translate genes into the proteins. Proteins plays all the functions and maintain the life. So the genes are the DNA with specific sequences that encoded the specific proteins. If some of these sequences of the DNA in a specific gene has mutations or miss one letter or with extra letter in that gene, so normally it will cause more function of the gene may not produce proper protein, and the protein may not be functioning. As a result, you'll have a disease. This disease is called a genetic disease. It's sometimes very hard to treat. And RNA is the middleman. It will translate DNA to the protein. Sometimes this middleman is very active, produce a lot of proteins that will also cause disease. Sometimes the middleman is lazy and produce less proteins. It also causes a disease. Also, there are some small RNAs like microRNA, link RNA. They also plays a role of regulating the functions of RNAs. If there's something wrong with this process, also cause human disease. Gene therapy involves at multiple levels. At the DNA level, you can use a normal gene to replace a malfunction gene or mutated gene and to restore the normal gene function. These days, you can also use gene editing to correct the mutated gene to restore the function. You can also do the regulation at the RNA level, use microRNA, siRNA, and link RNA to control the genetic functions to maintain normal genetic functions in biology. So the concept of gene therapy was proposed in 1972. It has been almost a half century since it first proposed. Over the period of this half century, we have some successful stories. For example, the first clinical trial was taking place in 1990s in a little girl with immuno deficiency. Normally we call them SCID children or bubble boys, bubble girls. They cannot live normally in a normal environment. So she received the first gene therapy with normal adenosine deaminase to restore the normal function. I think she's still pretty healthy. We also have some setbacks. For example, in 1999, the gene therapy trial cost the life of a boy named J.C. Gelsinger. This is caused by the side effects of the viral vectors. So this is actually a big setback for the gene therapy. It took another almost 20 years for FDA to approve the first gene therapy. So it's almost a half century from the concept was first proposed. So FDA approved this AAV-based gene for treating Leber's congenital disease to replace a protein called RP65. So it works well for a lot of patients and also doesn't really work equally well in some other patients. The therapy is very expensive for one eye, it's cost about $420,000. There are some other gene therapies that have been approved after this one. So all these therapies are very expensive, and there are over about 200 or 300 clinical trials with gene therapies right now is going on. So there's a really big progress in gene therapy in recent years. So this is a really exciting time for gene therapy, so have a good chance to allow us to treat patients that cannot be treated with a conventional approach previously. So I'm going to talk a little bit about some simple approach using non-viral vectors for gene therapy. If you see these 4 letters, you may think, okay, maybe this is something, is keep it simple and stupid. That's what normally used. In some other engineering approach, this is actually a very good approach for make things working. But in biology, in the gene therapy space, a biological system is very complicated. We'll have to have something much better than stupid systems. So that's what I changed. So I use this, keep it simple and smart. If you have simple and smart vectors, most likely you can reduce the cost for the production and also for cost for the therapy for treat human disease. So non-viral gene therapy has been investigated for many years, same as there's a virus systems. Mostly people use cationic polymers, lipids, to form nanoparticles with nucleic acids, and you can achieve targeting with these particles very easily. And the particles may taken by the cells and get in the endosomes, lysosomes sometimes trapped there. And a few particles can get out from the endosomes, but still DNA, RNA also trapped in the particles cannot be released in the cytoplasm. So that's actually a big challenge for non-viral gene therapy, how you can efficiently deliver nucleic acid payloads in the cytoplasm, maybe also in nucleus. So the challenge really for non-viral gene therapy is the endosome escape. How can you really facilitate endosome escape? There are a lot of other concepts has been proposed. Unfortunately, doesn't really work that well. So we proposed a concept about 15 years ago using the idea of incorporating pH-sensitive amphiphilicity and the reductive particle dissociation in the cytosol into a simple lipid structure to produce multifunctional lipids with the ability of pH-sensitive amphiphilic endosome escape and the reductive cytosolic delivery of the nucleic acids for high gene therapy efficiency. So, we're in the middle of a pandemic of COVID-19. One way to protect yourself to wash your hands with detergents thoroughly. The purpose of that is just to disrupt the membrane of the virus, so protect you from the viral infection. So with the same concept, you can make the lipids amphiphilic. As a result, you can, this kind of amphiphilic property will disrupt the endosome membrane. But if you don't control the amphiphilicity, your dissolved cell membrane will cause the cell death that could be toxic. That's why you cannot drink those Lysol or some surfactants to treat COVID-19. So it's the same purpose here. So what we need to do, we want to use a concept called pH-sensitive amphiphilicity. So these lipids will be neutral mostly at a normal pH. Once it's become internalized, get in the endosomes or lysosomes, pH become acidic. And then the lipids can be ionized and become amphiphilic. In this case, they were intact with endosomal membrane and release the particles in the endosomal membrane. At the same time, we also have this kind of reductive functional groups. We use a structure stabilizing the particle in the formulation, but this structure can be reduced in a reductive cytosolic environment, and then dissociate the particles to release the payloads in the plasma and can produce a high function. So we call this effects called a PERC effect. That's pH sensitive amphiphilic endosomal escape and the reductive cytosolic delivery. Based on this concept, we designed this multifunctional lipids. It has a very simple structure with protonated functional amino groups here. At the neutral pH, there will be neutral, not protonated as much as acidic conditions. It can also form particles with nucleic acid through charging interactions. And it has two lipid tails. This will introduce hydrophobicity of the lipids and also condense form stable nanoparticles. The key features, these two lipids are form a wedge. They are separate, located further away. So one form lipid bilayer. And we also have two thiol groups. These thiol groups can be oxidized to form disulfide bonds. These disulfide bonds will stabilize the nanoparticles during the delivery process. Once they get in the cytosolic environment, in the reductive environment, will reduce the disulfide bonds to dissociate the particles and release the siRNA. So this concept has been used by many others to design a polymeric or lipid other delivery system for nucleic acids. So actually works really well. So over the years, we have designed different libraries of these lipids. We found that this simple lipids called ECO works really well. E means ethylene diamine, that's head group. C is the cysteine residues, we have two here. And O is oleic acid. So it forms stable particles with nucleic acids through electrostatic interactions and the hydrophobic condensation of the tails. And also disulfide bonds can be oxidized by the air, and then they form a cross-linking as a result, form a stable nanoparticles. So unlike some other lipid-based nanoparticles, we don't use any other helper lipids. So we just use one simple organic compound. So I listed here, ECO. Just its alone can be form stable particles with a nucleic acid of any sizes. So here's some experiments we did earlier to show the concept actually working. So this is the concept, the multifunctional lipids form nanoparticles with the nucleic acid or be internalized by the cell, goes through this subcellular trafficking from early endosome, late endosome, pH drops over the time. And once pH drops, and these lipids will be protonated or ionized and become amphiphilic, and then destabilize the cell membrane and into the cytosol, and then the sulfur bonds will be reduced by the glutathione, and the lipids particle dissociate to release the payloads. So this is actually what we showed here. And 5 minutes, particles stick to the cell membrane, 30 minutes more, and some internalized. And 2 hours, you do see the overlap of particles inside intracellular compartments. About 4 hours, you do see a significant release of payloads in a cytosol. Of course, there's still quite a bit in the intracellular compartments, but a significant amount is released in a cytosol. I think this amount should be sufficient to perform biological functions. So recently we compare the efficacy of ECO for gene delivery with the popular viral vectors AAV. We use the GFP as a reporter gene. As you can see, actually our non-viral system produce much more significant gene expression compared to an AAV. So that's really exciting because previously people always believe viral systems are very efficient. It's much better than non-viral system, but we do show here our simple ECO particles can also mediate the highly efficient gene expression. Seems is at least better than this version of AAV here. So this is actually GFP expression in those retina cells. This is with our ECO. This is with a viral system. So we use this system to see if we can deliver a therapeutic gene to treat some visual genetic disorders. And the disease we're working on in the very beginning was the same disease that was approved for human use. That was Leber's congenital amaurosis. So with this approach, we loaded this RPE65 gene with nanoparticles, and we also use retina amine to target these nanoparticles to protein in the subretinal space called the interphotoreceptor retinoids binding protein. This protein transports retinoids to the RPE cells. So as a result, it will bring more nanoparticles inside RPE cells. Then they release the gene that express the RPE protein. So this is actually how we make the nanoparticle. It's much simpler and more cost-effective than these viral systems. So we have lipids, that's just very simple organic compound. So we use a small amount of thiol groups to modify the lipids to introduce the targeting group. We also use a space PEG. And then with this click chemistry, we attach these targeting agents to about 5% of thiol groups. And then we form nanoparticles with plasmid DNA, just with simple self-assembly. So just mix them together. So the particle size is about 180 to 200 nanometers. You can see these are the images of the particles. So we inject these nanoparticles into a mouse model of LCA disease. These mice have knockout RPE65 gene, basically it's blinded. So once we inject our particles into the retina space through subretinal injection, so after about 2 weeks or 1 week, so we can see the ERG signal. So that means the eyes response to the light. If we just inject with a simple PBAs, there is no ERG signal, and the mouse cannot see. There is no reaction to the lights. So here we shows the actual protein expression in the RPE layer with controls. We don't see anything here. So this shows that our non-viral system works well for treating LCA disease. So the therapeutic efficacy lasts at least for 4 months after single injections. So we didn't really do a long-term follow-up after this. So next one, we want to see how we can deliver genes that AAV cannot do, because AAV has a small loading capacity. RPE65 is small enough to load in the AAV. For some other retinal genetic disorders like Stargardt disease, it has mutations in some big genes like ABCA4 gene. This gene is too big for AAV to load, to transfect the cells. So Stargardt disease caused by the mutation of an ABCA4 gene in photoreceptors. So this mutation caused a loss of central vision initially, and gradually you lose, patients will lose eyesight completely. So we thought, maybe we can use our ECO to load this gene to transfect gene into photoreceptors to see if we can prevent the progression of this disease. So we formulate ECO nanoparticles with ABCA4 plasmid DNA. This plasmid has a size about 12,000 base pairs. Definitely this is too big for an AAV. So we inject these nanoparticles subretinally. We do see significant expression of the therapeutic gene in the photoreceptors. We did two promoters. We use the different promoters to control specific gene expression. For example, if we use the CMV is a promoter from viruses. It's not very specific. It promotes gene expression in almost all the cell types. For rho promoters, is specific for photoreceptors. As a result, you can see we only see expression in the photoreceptors. With CMV, we do see gene expression in RPE and some other layers. So with rho promoters, we have highly specific gene expression. This will reduce the non-specific side effects of this ABCA4 gene. Wild type, of course, you can see has much higher expression. But anyway, we can deliver whole ABCA4 gene with our ECO nanoparticles. So efficacy-wise, we followed up some other biomarkers. So this is after single injection in mice with ABCA4 gene knockdown. So this is a model of Stargardt disease. So with the rho promoter, we do see a significant induction of a biomarker of Stargardt disease A2E in some eyes. For control eyes, you can see this signal is pretty big. After this one, we do see reduction. Some of this actually prevent completely the accumulation of A2E. So this is a group of mice treated with nanoparticles of a CMV promoter. I'm sorry, this is a typo here. CMV promoter. You can see have some efficacy, but not as great as the rho promoter. So the overall with a rho promoter and the ECO nanoparticles, we do see around 70% reduction of A2E, and for CMV promoters, it's about 10%. So that actually shows that our ECO nanoparticles works really well to transfect functional genes in a disease model. So this is the result after 6 months of a single injection. And we also followed the gene expression about 8 months after injection. Seems so we still have stable gene expression in mice at 8 weeks after injection. This is a messenger RNA. And we also did a histology. You can see at 8 months after injection, we still see significant expression. For CMV, actually, we don't really see a whole lot. So that shows that our simple ECO nanoparticles is able to deliver small genes and big genes to treat visual disorders. So we also are trying to do some other disease, see if we can also use this one to treat other disease. Gene editing is also promising to treat human disease. CRISPR-Cas9 and some other gene editing technology right now has been explored for gene therapy. So we want to see how our ECO works to mediate the gene editing. We compared with one of the commercial available lipid carriers, lipofectamine. As you can see, these cell lines has a strong expression of GFP. GFP we use as a reporter gene. Lipofectamine does have some knockdown effects, but ECO works much better. You can see ECO mediate almost complete knockdown of GFP. The lipofectamine has only little effects. So that means the ECO is also highly efficient to mediate gene editing with high yield here. So we also test the ECO system for gene regulation with RNAi in cancer therapy. This approach is forming nanoparticles with siRNA is very simple. All the RNAs, DNAs we use are chemically unmodified. So this will reduce some other impacts from chemical modification. So we can also modify these ECO nanoparticles similarly by conjugating targeting groups with a PEG spacer to a small amount of thiol groups. Normally we conjugate modified about 5% of the thiol groups. And then we mixed with siRNA about half hour, we can form stable nanoparticles you can see here. So our particles can be stored for at least 12 months without changing any features here. So it has a very good high stability. We don't really use a very high amount of lipids like some other systems do. In the lipid amine to phosphate ratio is only 8. So we test the particles for treating some metastatic disease. For example, triple negative breast cancer is a highly invasive type of breast cancer, causes high mortality rates. So we want to see if we can regulate some genes associated with metastasis. In this case, it's a beta-3 integrin. This is a switch of EMT. TGF-beta induced the EMT. So we test these particles in animal models with lung metastasis. So we treated particles with just non-specific siRNA and a specific siRNA with non-targeted nanoparticles and targeted nanoparticles. We inject every 5 days systemically at a dose of 1.5 mg per kilograms. You can see after 9 weeks, the control groups start to form a mass. Treated groups has no mass formed. So we sacrifice the mice and we check the mass formation. These control groups, you don't really recognize this is as a lung because tumors are over the lungs, but the treated groups has nice clean lungs. That means our particles is effect to deliver RNAs to regulate oncogenes to prevent metastasis. We also modified the nanoparticle formulation to introduce the function of the nanoparticles to sense the environmental change to enhance RNA or DNA delivery. In this case, we introduce a pH-sensitive hydrazone linker to allow more efficient endosomal escape. So particle formulation is very simple, same as what we showed before. We modified small portions of thiol groups and they form nanoparticles with DNA, RNA automatically. And once these particles is injected into subjects or recognize tumor regions or internalize. And once internalized in the endosome, pH drops and low pH will hydrolyze these hydrazone groups and expose the nanoparticles. And nanoparticles, as you know, is pH sensitive. So pH sensitive amphiphilic property will destabilize the membrane and release the core particles, and then core particles will release siRNA in the cytosol. So without these hydrazone groups, this PEG layer may hamper the endosomal escape process. With this approach, we will enhance the endosomal escape more effectively. So actually it works really well. This is examples here. This is non-targeted nanoparticles. We do see uptake of the particles in the tumors. Tumors are labeled with the fluorescence probes. But targeted nanoparticles will have much higher uptake. So this is the quantitative results, shows that targeted nanoparticle has much higher uptake at 24 hours than the non-targeted nanoparticles. That means targeted nanoparticles can facilitate tissue tumor uptake. But for gene silencing efficiency, without this pH-sensitive linker, actually there is a big difference. This is a quantitative result after injection at different days. So first day we do see, the particles with pH-sensitive space has significant reductions. And it's also true with some other nanoparticles. But for some other nanoparticles without the hydrazone linker, for example, a PEG, RGD targeted PEG, ECO nanoparticles, you start to see the comeback of the gene expressions. That means silencing efficiency is not that high. But this targeted nanoparticles with hydrazone linker actually maintain significant gene silencing for at least 7 days after single injection. So this is actually reflected in the bioluminescence imaging. You can see this is a non-targeted. This is non-targeted with hydrazone seems a little bit than a non-targeted. This is targeted without hydrazone, but targeted with hydrazone linker works much better than just a non-target, without the hydrazone linker. That means the introduction of this pH-sensitive linker significantly enhance the gene silencing efficiency because this kind of environmental responsive release and the delivery of siRNA in the cytosol. So this is some further characterization of nanoparticles. You can see without this hydrazone linker, the surface charge of particles did not change at different pH. And with hydrazone linker, actually, we do see particle has more positive charges at acidic pH. So this kind of environmental sensitive property of this hydrazone linker, we also call this dual pH sensitive nanoparticles, result more uptake of, more release of siRNA inside the cytosol. And you can see as a result, we have more significant gene silencing of a reporter gene. You can see this is a dual pH sensitive or environmental sensitive ECO nanoparticles. With RGD is up here, it's only about 40 or 30%. And with this dual pH sensitive nanoparticles, actually we have at least 80% gene silencing efficiency 3 days after the treatment. So, and then we tested in vivo property of these nanoparticles for therapeutic purpose in a drug-resistant tumor model. So I don't want to go through all the details here. I'll just show you some results here. So this tumor model is a drug-resistant triple-negative breast cancer. It's highly invasive. Without treatment, you can see the tumor is pretty big and tumor grows really fast. If you just treat the tumor with non-specific nanoparticles and the drug here we use is paclitaxel, you can see tumor grows faster. Seems like even faster than the control groups. And if you just treat the tumor with siRNA nanoparticles specific to a protein that are responsible for the drug resistance, actually, we do see stabilization of the tumor, not much change of tumor size. With a combination of this SI-EI4E nanoparticles and the drug, we do see actual regression of the disease. So that means these particles actually works really well to mediate gene silencing to sensitize the drug-resistant cancer cells to therapy. So, so this is the summary of what we have done with these very simple lipid nanoparticles. The lipids, as we showed at the very beginning, was designed with the function of pH-sensitive amphiphilic endosomal escape and the reductive cytosolic delivery. It shows that this lipids actually works really, really effective for deliver nucleic acid of any size, from small siRNAs to big genes. So the particle formulations process is very simple and straightforward. And unlike some other gene therapy process, the production is very expensive. This will be relatively less expensive. So we also show the particles works well for gene editing and for gene regulation. So we try to move it forward for clinical development in next stage. Hopefully, we can use this to deliver genes to treat disease, excuse me, some other gene therapy cannot treat. So, and then I want to thank my team members for all the work. So this is Da-san and Wendy. They actually both worked on the gene therapy projects, and Amida and Peter and Ryan, and worked on the siRNA therapeutics. Thank you.